Hello, my name is Carmel Heiland, and I will be discussing with you erythropoietin detection from dried blood spots for anti-doping purposes. Dried blood spots, or DBS, have been researched for several years for substances that are prohibited in sports, and the challenges and advantages have been discussed on several platforms. But there are still many challenges that need to be addressed, especially when it comes to EPO detection. The aim of this study was to practically evaluate the performance of EPO detection from dried blood spots. For this, we looked at the sample collection volumes compared to EPO detection to find a proper balance, the detection limits that can be achieved from such small volumes, and the stability of EPO in dried blood spots compared to urine, which is the most common matrix for doping control. In this study, a whole blood pool was spiked with various erythropoietin receptor activators, or ERAs, and the blood was spotted or pipetted onto collection cards and dried. The spots were then cut out, and the blood was extracted and immunopurified, and the EPO was immunopurified using the MAIIA EPO purification gel kit for blood. This kit is commonly used for immunopurification of serum and plasma samples, but the method was modified for the purpose of dried blood spots by concentrating the sample down to a final volume of 15 microliters. The immunopurified samples were then analyzed with sarcosyl polyacrylamide gel electrophoresis, which separates the EPOs and ERAs based on their molecular weight. These EPO proteins were then subjected to a single Western blot procedure where the detection antibody was biotinylated. It's important to have a balance between the amount of blood that can be collected from an athlete and the ability to detect EPO in the lab. We evaluated this in two ways. For the first, we looked at the volume of whole blood that was pipetted onto one spot. Here we looked at 20, 40, and 60 microliters of blood. You can see that endogenous EPO can be detected from 20 microliters. However, the signal intensity is still faint, and 60 microliters of whole blood gives a better detection of endogenous EPO. We also looked at the number of spots that could be immunopurified at the same time in order to increase the sample volume. We looked at one spot which had 60 microliters, or two spots that were immunopurified simultaneously, which gave a total of 120 microliters per sample, or three spots that were immunopurified at the same time, which was 180 microliters per sample. You can see that there is a linear increasing trend in the signal intensities as the number of spots increase. However, collecting 120 or 180 microliters of blood is not practical, especially when blood also needs to be collected for other analyses. In this case, 60 microliters of blood from one spot is an optimal volume to be collected and from an athlete and for EPO detection in the lab. We also looked at the detection limits of 3 ERAs, CERA, EPO-Fc, and NESP. We looked at 4 different concentrations in a serial dilution, as you can see from the table. You can see from the image that the 3 ERAs could be detected down to the lowest concentration, concentration 4, in 60 microliters of whole blood. If you look at the concentrations in the table, they're calculated for picograms per 1 milliliter of whole blood. This is approximately equivalent to half a milliliter of serum, and in this way, we can compare the detected concentrations from our experiment to the World Anti-Doping Agency's minimum required performance levels, or MRPLs, that have been published for EPO detection in anti-doping labs for serum. Here you can see that CERA and EPO-Fc have about 5 times and 2.5 times higher concentration than the MRPLs from WADA. However, the concentration that we detected for NESP is quite close to the MRPL. It's important to know how sample storage prior to analysis can affect EPO detection. For this, DBS samples were prepared and stored at room temperature for 48 days. The same was done for urine, stored at the same conditions. You can see from the image that after 48 days at room temperature, EPO was well detected, however, in dry blood spots. However, in urine, the signal intensity decreased significantly and the samples were quite degraded, making EPO detection quite difficult. Therefore, the EPO was stable in the dry blood spots for the conditions that were tested. For the future experiments, we would like to continue to look at the detection limits for the ERAs, particularly for recombinant EPO, and also compare polymer and filter paper supports for whole blood. We would also plan to collect and analyze samples from administration studies for recombinant EPO, but also looking at endogenous EPO on its own. Thank you for listening.